Hey, welcome back, SEA-PHAGES, to another fun week of serial dilutions. I know everyone loved it 2 weeks ago. We're going to do it again, just a little bit different though. So buckle in for that. So Titer MTL week, MTL standing for medium titer lysate, is going to be found in your lab manual pages 52 to 53. Not really a long protocol, but some things are different. So make sure we're paying attention because we're doing some critical thinking this week, and it's all dependent on you guys. So important stuff. So briefly, let's talk about last week. So we collected our lysates last week. Again, lysates are a concentrated phage solution. So we are going to be using this for the rest of the semester moving forward. So again, we are going to, like I mentioned, every week for the last 3 weeks, we want to be able to characterize our phage. So with that, we're going to do a quasi-measurement of activity. So we're going to determine the concentration or titer of our lysate by performing this week a serial dilution and then a plaque assay for each dilution. And then next week, we're going to count the number of plaques formed on those dilution plates to get our titer count. So titer is going to be measured in a unit of PFU, so plaque forming units per milliliter. So PFU per ml is going to be our unit for our titer. We're taking into account in this calculation the volume used to plate that particular plate, so 10 microliters. We've been doing this since last week. And the dilution used to generate that particular plate to calculate our titer. Again, this is a estimate, right? I've mentioned this every week for the last 3 weeks. This is an estimate of the number of phage particles in your sample. It is not a measure of every single phage particle. It is just an estimate because it is only measuring those that are infectious, the ones that are able to infect bacteria, lyse them, and then form plaques. Okay, so just an estimate, but this is the best we can do in this particular setting. So this is the equation that I've been throwing around a couple weeks now, but next week I promise you get to use it. So bring your calculators. We're going to do some math next week. But this is our titer calculation we want, and it's heavily dependent on this number PFU. So our countable number of plaques. This week, our goal with our serial dilution is to produce a countable number of plaques on a plate. Our goal is 20 to 200. If it's under 20, it's a little bit inaccurate, our titer calculation. If it's over 200, it gets a little bit difficult to count. And in that case, you run into this human error situation where you're counting them wrong, so your titer calculation is wrong because of human error. So to get this countable number of plaques, it really depends on your results last week. So to do the serial dilution, again, we're diluting serially our lysate, but how far you diluted out to get this countable number is very dependent on what results you got last week. Okay, so what plate you flooded last week. So let's elaborate on that a bit. So consider, like, everybody flooded a webbed plate to produce a lysate. A web plate, just so we're on the same page with that, is a plate that is heavily concentrated with phage. It has a lot of plaques and bacteria. It's evidence of reproduction, amplification, lysis, all of those happening over and over and over again. You have a high concentration of phage in that plate. The dilution concentration of whatever plate you flooded will determine how far out you are flooding out. Okay, so for example, if your web plate, if you flooded a plate that was webbed and it was your 10 to the negative 1 dilution, that tells you that 1/10 of your original inoculum, of your original phage particle sample, produced such a web plate. Okay, it was not immediately lysed, despite the fact that it had quite a bit of phage in it. It wasn't immediately lysed. So you do not have to, dilute out very far to get a countable number, okay? So if your web plate was 10 to the negative 1, negative 2, you only have to dilute out a little bit further before you start seeing individualized plaques, okay? On the other hand, if the plate that you flooded was your 10 to the negative 4th, if your web plate that was produced was a 10 to the negative 4th plate, that tells you have a little bit a high activity in your particular phage. So 10 to negative 4, just for context, had 1/10,000th of your original inoculum. Not a lot of phage in there, but it still produced, it was still so highly concentrated, okay? So you're gonna have to dilute it out a little bit farther to be able to get a countable number of plaques in that particular case. If such a tiny amount of phage in your dilution produce so much, so many plaques, so much phage evidence, you're gonna have to dilute out a lot farther before you start getting individualized plaques. Hopefully that makes sense. But think about this before you come into Open Lab, to have a little discussion with your partner, based on our results, how should we move forward? Okay, have a plan of how you're going to, how far you're going to dilute out. Okay, so if you're between like 10 to negative 3, negative 4, if that was the web plate that you flooded last week, you're going to have to dilute out a little bit farther, to around 10th to negative 7th or negative 8th even, before you start seeing plaques. But you only have to plate 5 of those. So what I mean by that is you are going to be doing this dilution. You're going to be diluting out all the way to like the negative 7th from your original inoculum. This week it's going to be your lysate. But you only are going to plate the last 5. The reason for that is that these first 2 are probably going to be just completely lysed or heavily webbed. But around like maybe 4th, 5th, or even 6th, you'll see countable number of plaques. But we plate 5 out of an abundance of caution. Okay. On the other hand, if you flooded a plate that was webbed, that was 10 to the 0, 10 to negative 2, that tells you don't have to you don't have to dilute out very far before you start seeing any countable plaques. So you're only going to dilute out to around like negative 5th, and you're just going to plate all of these, again, out of an abundance of caution. But sit down with your partner or, over some kind of chat, text them, FaceTime them, Zoom them to discuss how you're going to move forward with Open Lab this week. Come with a plan, come knowing, based on my results, based what plate I flooded, if it was a web plate, if you flooded a plate that just had a high number of plaques, it might not have been webbed. Think critically, how far out do you have to dilute before you start seeing a countable number of plaques? And obviously the Open Lab TA, your Restation TA, we can help you, but come with a plan. Don't just come in completely blank thinking that we're going to tell you what to do. This is data analysis, critical data analysis. You need to look at your own data and think about how to move forward. And we'll be there to guide you, but this is a time when you really should be starting to kind of think for yourself on how to move forward. Okay? We're building scientists here. So let's talk about procedure. So same procedure for the most part in terms of serial dilutions as 2 weeks ago. So you're going to get 5 Petri dishes because everyone's going to plate, every group is going to plate 5. It's not individualized, it's groups. You're going to do 5 Petri dishes. They should be matching the labels with the appropriate dilution that you are plating. So what I mean by that is that if you are, diluting out all the way to negative 7th, but you're only plating these last 5, you should be labeling all of your tubes, your culture tubes and your Petri dishes so they match these 5 dilutions, just as an example, okay? We all know what to label on the Petri dish, our date, initials for both of you, for your groups, or if it's a group of 3 groups, the content and the dilution amount, very important, okay? You're going to label your culture tubes also to match the appropriate dilution, okay? But for microcentrifuge tubes, you're going to get however many you need to however far you're diluting out, and you're going to label all of them appropriately, okay? So in some instances, some groups might have more microcentrifuge tubes than they do Petri dishes and culture tubes, because you might not be plating all of your entire dilution. You might be plating a portion of it, okay? But you still need to get all of the microcentrifuge tubes. You need to label them with the dilution amount, just so we're sure moving forward, okay? So this is where there's a little bit of a difference from the last serial dilution. Every single microcentrifuge tube is going to get 90 microliters of phage buffer, and you're going to be adding it aseptically to all these tubes, okay? All of them get 90. And this is just going to be a little bit of a refresher on how to get microcentrifuge tubes out of the container, because it is a sterile container, and we saw some not so aseptic technique last time we did serial dilution. So this is just to clear it up. Okay. So you're working within the aseptic zone. You're going to pour some of those microcentrifuge tubes onto the lid. Okay. You're going to close those tubes in the aseptic zone. So it's not directly in the fire because they are plastic, but you're going to close them in that heat so any microorganisms can be raised by like the raising heat. Okay. You're never putting your germ-infested hands into that microcentrifuge tube container, which is sterile, because otherwise you will contaminate it for everyone. Okay. So don't be doing that. And this is just a refresher on how to do the serial dilution. This is the exact same thing, adding 1 part of your inoculum to 9 parts of your buffer, but let's just, as a refresher, watch it. So again, this is the exact same thing that we did 2 weeks ago, okay? But the source of our phage is what's different here. So previously, when you did your cell dilution, you took a sterile wood stick, you poked a plaque, and you swirled it. It's a phage buffer. Not the case this time. Okay, your lysate is going to be your 10th this year. That's going to be your original concentration of phage, and you're going to be diluting that all the way through. Okay, so to do that, we're going to be aseptic here. You're going to transfer 10 microliters of your lysate into your first microcentrifuge tube, which has 90 microliters of phage buffer in it. Okay, you're going to transfer 10 in. You're going to, flick the tube to make sure we're mixing. You should be doing that every single time to, make sure that you are mixing up that solution well in that phage buffer. Okay, the final volume, once you do that, that's going to be 100 microliters in that microcentrifuge tube. This is going to be your 10 to the 1, -1 dilution. You have just diluted your lysate so that this centrifuge tube now has 1/10 of that original phage concentration. Okay. Then you're going to transfer 10 microliters from this 10 to the -1 dilution into your next tube, which has 90 microliters in it. You're going to flick it to make sure you're mixing up that entire mixture. You're going to be making sure you are switching pipette tips between each dilution. If you don't do that, you are not serially diluting it accurately. Okay, so this second tube here, you just transfer 10 into it. It's going to have 100 microliters in it now. This is going to be your 10 to the -2 dilution. And now this tube here on the right, this has 1/100th of your original phage concentration that was in your lysate. Okay. You're going to continue doing this until you have reached your final dilution. So for every single one, you're going to be transferring 10 microliters into the next tube with a phage buffer. You're going to flick that microcentrifuge tube to mix it. You're going to get a new tip, and you're going to do it again. Okay, all the way out to either 10 to the -7th or -5th, or whatever dilution you are diluting out to. Hopefully that makes sense. The final tube, the very last one, that's going to have 100 microliters in it. Okay, you're just transferring 10 to 10 to 10 to 10. Okay, So these next steps are identical to what we did 2 weeks ago. So you're going to take 10 microliters of each dilution that you are going to be plating, right? Whatever 5 dilutions you're plating, you're going to take 10 microliters of them. You're going to add them to 250 microliters of your Gordonia in culture tubes that are appropriately labeled. You are going to let that inoculate for 10 minutes undisturbed, okay? Then you're immediately going to add 3 milliliters of top auger to those culture tubes, and you're going to immediately plate it. And you can do the swirling this time, okay? That's allowed this time. You can just really get it around. Do this one at a time. I recommend this again, because if you are adding top auger to all of your culture tubes at one time, by the time you finish adding it to your last culture tube, your first one will be mostly solid. So do this one at a time. Add your top auger, plate it. Add top auger, plate it. Top auger, plate it, okay? And make sure everything is labeled before you start doing this. Even if you think you're gonna remember which one is which, and you say, oh, I'll just write the dilution after they're solidified, you won't remember. Just do it before, okay? Save yourself some time. So that's it for the protocol this week, but let's remember some key things move forward. One, generally you should be coming into Open Lab having some sort of plan for how you are going to be diluting, how far you're going to be diluting. Have a conversation with your lab partner so you both understand what you're going to be doing this week, okay? Make sure that you are switching pipette tips each time you're transferring tubes. Make sure that you're mixing those microcentrifuge tubes after every time that you transfer. Make sure also that you're shaking up your bacteria before you use it. That's been a common issue. If you see a little tiny orange pellet at the bottom, that's bacteria. Shake it up so it's in the solution. Plate your top auger bacteria phage solution immediately onto your plate. And then continue practicing good aseptic technique, right? Don't be contaminating everyone's microcentrifuge tubes. We really want to see some really honed in aseptic technique this week. And then make sure you and your partner are going to be practicing good social distancing still. You're working in a group, in a pair, in a group, whichever, but you still have to be socially distant because of what's going on, right? You're working together, but you're going to have to be as distant as you can be while doing that. So if that means, one of you is pipetting something and then steps away and the other person comes in to do some other portion of the pipetting or something like that, just make sure that you are socially distant while doing your work. And as always, please make sure you are labeling everything, just so there's no mix-ups. Okay, we're paying attention to what we're pouring into what tube, to what plate, so that the label matches every time. Okay, I believe in you guys. This week, our open lab is again 2 hours. So make sure that you and your lab partner are signing up for a 2-hour lab session, okay? If you have any questions, as usual, you can contact me, you can contact your TA, we can answer anything for you moving forward. But best of luck this week. It'll be a fun time.